This is a production of Cornell University.
So thank you so much for giving me an opportunity to talk to you guys about the research that I'm doing in my lab. So as mentioned, I'm really interested in how stress is changing architecture. So if we look at the world map of problems that are affecting agriculture in general, you can see that a lot of problems are indicated by the blue dots, all kinds of shades of blue dots. And those are indicating problems associated with water stress. So either we've got too much water, too little water, or wrong kind of water. So we've got drought, waterlogging, or salt stress. And my approach of kind of, or like my thinking, how I grew up intellectually, is we can solve those problems by two different things using genetics. So we can either diversify our agriculture, so look at the plants that are thriving in those environments that are really extreme, and learn how we can domesticate those things. So don't be fooled by my Photoshop skills. This is a desert rose that is a native to this small island of Socotra off of the coast of Saudi Arabia. And it's really thriving in a very, very extreme heat and drought stress environment. And we can think about like, okay, how can we make something that we're interested in as humans? Or inspired by the work of Nikolai Vavilov, we can look at the plants that are closely related to our crops that we're growing in today and look at the close relatives of those crops and how are they dealing with all kinds of stresses. So look at the variation within the species and very close to the species that we're normally growing. So within my lab, we're working on a couple of plants, and most of them are really super tough. So I'm really inspired by super tough plants. Arabidopsis thaliana is actually also a super tough plant, especially if you're not trying to grow it in the lab. Because if you're growing it in the wild, it's thriving between the pavement plates on the street. But we're also studying wild tomatoes, Solanum pimpinellifolium. I'm going to talk a little bit more about that. So Solanum pimpinellifolium is one of the closest relatives, and it's native to west coast of South America. So that's an environment that has a tremendous range of different environments. We're also studying tepary bean, Phaseolus equitifolius, which is a very close relative to common bean. But this one is native to southwest of the US and northern Mexico. So again, a very dry environment. This plant has been used by indigenous communities in their dryland agriculture. So there is a tremendous variation over there. And it is, you can see in here, so it's called equitifolius because it has very narrow leaves. And the leaves compared to the common bean, you can really see it's almost needle-like, especially if you're looking at the variation in the wild relatives. And then we're also studying cowpea. So cowpea is Vigna unguiculata. It's extremely popular crop in sub-Saharan Africa and Asia. It's grown widely by subsistence farmers, and it's also drought tolerant because nobody over there, or hardly anyone, has irrigated agriculture, and the plant is thriving. Yet, this cowpea has leaves as big as my hands. So very different strategies of dealing with drought stress. And we're hoping to learn what are the genes underlying those different strategies so we can later on figure out like best strategies to kind of like pyramid all of those genes together and use the strategies that are hopefully complementary to produce the super crops that we need in the future. So what is the general strategy that we're using? So I'm really starting with the phene of interest. And the phene, what is the phene? So a phene, relation of a phene to a phenotype is the same as a relationship to a gene to the genotype. So phene is an atomic component of the whole phenotype. So I've got a phenotype today, this is what it is, but this pink sweater is a part of my phene. So if you've got a phene of a plant, for example, a height of the plant so many days after germination is a phene, or how many, like the size of the leaves or the length of individual components of the root system architecture is a phene. And the idea is that we can map things that are genetically very complex, like stress resilience, which has many genes that are contributing to it, by focusing on things that are a little bit more heritable and a little bit closer genetically, a little less complicated on a genetic level. So we're dissecting the plant to like anatomic components that we can observe, phenes. We're looking at, is this phene at all changing in response to stress? And that's really where I'm interested in. Because if it's, breeders typically don't like plasticity. They like the trait to be like as fixed as possible. But those traits are always the same in all of the environment, which is great. But we're missing out on a lot of plasticity that we're not integrating into the current breeding programs. And of course, because we're using them for the genetic studies, we're looking at the heritability of individual phenes, okay? So once we've got that figured out, we're moving into the population screening. So we're doing a lot of phenotyping, high throughput. Most of the phenotyping that we're trying to do is high throughput. Some of it is still high dedication. I'm going to talk a little bit more about that. And we're using forward genetics to identify which genes we really are associated with those stress resilient responses. Once we are at the gene of interest level, we're validating it using reverse genetics, identifying molecular context to better be able to translate it between individual kingdoms. And also at this stage and at this stage only, we're looking at the relationship between our phene of interest and the overall plant productivity and the stress resilience. Because if we're looking at the correlation between my root length or number of lateral roots, stuff like that, and the plant yield or the plant salt tolerance, in here at the natural diversity panel, it's similar as if I would be looking at the phenes that are predictive of your academic impact index, right? So we are different. We are having those academic impact indexes achieved in a very different ways, and therefore looking at the simple correlations is not going to be very informative. Only if I'm going to clone one of you that is one of you in general, and then mutate a specific genes, am I going to be able, which gene is really contributing to your academic impact, okay? All right, cool. So moving forward, so again, I'm interested in how stress is changing plant architecture. So in here we see a plant, Arabidopsis, exposed to increased concentrations of salt stress. So you can see that the plant is growing smaller. My question really is, how different is it growing? So what we have done during my PhD and after, we have set up a very system models. And you can think about the root to shoot ratio. Is the shoot impacted to the similar degree as my root when I expose it to the salt? And if not, and we know that, for example, drought stress, the plants tend to get rootier. Therefore, there's a change in architecture. So the question is, are there also any changes in the root system architecture, right? So is my main root being impacted much more severely compared to my lateral root development? And we know that those tissues developmentally have a very different identities. So they should also have a very different response to stress. So we have set up those models, and all of those papers are described. And we have used initially the population of Arabidopsis thaliana, consisting of 250 or 350 accessions collected from all over the world, although this population is a little bit outdated, I have to say. We have collected it early in the 2000s, and it's very Eurocentric, as you can see on the positions of those dots. In the meantime, we have expanded the population of Arabidopsis accessions, and when I was doing my postdoc in Saudi Arabia, we also have collected some of the Arabidopsis from there, and I have colleagues that have collected Arabidopsis from all over the Africa. So there is really a new layer of really interesting evolutionary history that is coming. So this is an example of how we have set up the work. What I really want to focus on today, especially to this audience, is not talk about Arabidopsis. Let's talk about something that is much more interesting. So this crop is Solanum pimpinellifolium, a wild tomato. I have introduced it to you earlier. So we have, Solanum pimpinellifolium is the closest relative to cultivated tomato. And we have ordered 220 accessions from the TGRC, which we have extensively genotyped. We also have developed a reference genome, and also Faye at Boyce Thompson Institute have sequenced some of the pimpinellifolium genomes. But you can see there we've got a wide range of pimpinellifolium accessions that are really spanning wide range of environments across the west coast of South America. And initially, we really started working on the pimpinellifolium because we were interested in this like almost mythical salt tolerance of tomatoes growing on Galapagos Islands. And we thought, those tomatoes are really growing into the sea breeze. They must be salt tolerant. So we have screened a bunch of Galapagensi and Cheesemani. And by accident, TGRC has sent us one seed of pimpinellifolium, and that outperformed everything else. And then we're like, okay, maybe it's not Galapagos specific. Let's have a look at the pimpinellifolium. So we have screened those TGRC accessions in the field in Saudi Arabia when I was during my postdoc. And the pictures that you're seeing are the pictures that are collected in the Australian plant accelerator facility, which is another phenotyping facility. But we're definitely able to produce those kind of pictures or grow tomatoes until those stages within our new facility at the BTI. So it's exciting. So this paper is done, wrapped up. But me being me, I really like to look a little bit more detail. And the shoot architecture, I'm realizing the more I work it, even though a lot of people are working on the shoot-based traits, if you look at it and if you have to automatically extract all of the, how many branches does the plant have, how many leaflets do we have, it's actually quite hard on the computational level because you've got all of those occlusions. Right, so shoot architecture is more complicated. So what I have done is I have screened the entire diversity panel for the root system architecture. So how are we doing it? We're germinating the seeds on the agar plates for 4 days, and then we transplant them to 2 agar plates containing no salt and 100 millimolar salt. And that's then resulting into a lot of images that have to be quantified. And this is one of the examples of high dedication phenotyping methods, because we have tried to have multiple machine learning methods to track the root system architecture, but as soon as the roots are overlapping, the computer is going like, I don't understand what's happening in here. So, but I have done this screen just before the pandemic, which was perfect timing because we have spent all of those months being anxious and tracing root at the same time. So you've got some mindless work that you can put your energy in. So, and that work, I just started at BTI when I finished this screen. So Haley, who is a technician, helped with tracing all of those thousands of root images. So from all of those 220 accessions, what we have identified is we've got 2 accessions that we really wanted to dig a little bit more into. So I present you a lateral root maker and the lateral root elongator. So those 2 accessions under control conditions are growing very similarly. You wouldn't be able to tell a difference between them. But under salt stress, one of them keeps on producing new lateral roots. They don't grow very long, but they keep on emerging. And the other accession is producing longer lateral roots. So the lateral roots that are there, they are keeping on elongating, but it doesn't really produce any new lateral roots. So we're like, okay, this is really cool. We had the data only from like every day or so. So we wanted to image them on a more higher time density. And we used this Spyro machine, which is a 3D printed machine that you can put the plates in there, and the thing is basically rotating every hour or so, and we can take images of the growing roots every hour, which leads us that we can identify at which time point are those 2 accessions starting to really grow differently. So we have in here the lateral root length of control and salt stress. Salt stress is in red, control is in blue. And you can see that our lateral root elongator is showing no difference, and that our lateral root maker, it's really inhibited in the lateral root length of individual lateral roots, okay? So in here we see a difference, and here we see no difference. And then we can see a similar trend when we look at the lateral root growth rate. Then when it comes to when are the lateral roots popping up, we see in our lateral root maker that the lateral roots kind of keep on popping at all the time points. There is a little bit of a change, but if you see at individual like lines, there are kind of like, you see blue lines and the red lines all over the place. Whereas in our lateral root elongator, you see that it takes much longer for the lateral roots to really start popping up, right? So that kind of gives us a little bit more time points for conducting RNA-seq assay. What we have also done, or actually Mariam, so Mariam Ramati-Ishka was a postdoc in my lab. She just left because she got her own position at Eastern Illinois University. But what Mariam did is also cross those lines, because we were wondering like which of those traits is dominant. Are you more, is making new lateral roots or elongating lateral roots more dominant? So she generated an F1, and what we've seen is that both of those traits can be inherited. So we can really make a super root. And I have to admit that both of those lines are already salt tolerant compared to all of the other lines. So we're really able to kind of staple the salt tolerance on top of each other, which I think is pretty cool. So out of this F1, we did a bulk segregant analysis in collaboration with Zhang Junfei at BTI. We also have done the genome-wide association, and we also have done an RNA-seq, and that was in collaboration with Andrew Nelson and Liang Yu at BTI. And so basically, we've got hundreds of genes that are coming out from each of those analyses. What we have done is really look at what is the overlap. And so the overlap in here, we've got 4 genes that are overlapping between 3 of the techniques that we used. 2 of them are unknown. One is really like unknown. The other one is DUF something, and DUF stands for domain of unknown function. So we know that the domain is repeated over the genome, but we don't really know what that domain is. So we are slowly working on making that domain of unknown function domain of unidentified function. So we're carving the DUF into a diff. But the cool story that we also observed is this gene that was overlapping between our RNA-seq data and the genome-wide association. It's a SWEET12. So SWEET12 is a sugar transporter, and it's actually a sugar exporter. Sweets have been so far really studied in the fruit development world, so reproductive biology, but not much of it has been really studied in the root system architecture. And we're like, okay, so this is really cool. Let's see what this sweet is doing. So maybe, and I have been thinking maybe, with the similar to fruit development, in terms of the lateral root development, what we also can see is that there is a developing relationship of sink-source relationship, and many of the sugar transporters or even ion transporters or other things can act in a very similar fashion when you're developing a fruit or when you're developing a root, because it's just this very initial stage of establishing sink-to-source relationship. So in tomato, we have not yet made the mutants. I just have been on the conference last week on the plant membrane transporters, and there was one person working on SWEET12 in tomato, and again, they have been only looking at the fruits, but they have promised to share the seeds with me so we can have a look at what's happening in the roots. So that's exciting, but that's coming up. What we have done is, again, going back to my Arabidopsis roots, it's a weed, but it's a helpful weed. So what we have done is we looked at the SWEET12 and 11 phenotype in Arabidopsis roots. At the same time, we also looked at all of the other sweets that are expressed in the roots based on the single-cell RNA-seq data. So SWEET3, we didn't really see much of a phenotype in here, but for the SWEET12, we observed that when we have a tDNA insertion, we're still figuring out, is it really a loss of function because the qPCRs are ongoing. So we really observed that those mutants are developing larger root system architectures under salt stress. And if we knock out both SWEET11 and SWEET12, we also have a significant effect over there. At the same time, we're like, okay, well, there are all kinds of transporters that are expressed in the root, specifically in the pericycle. So pericycle is the tissue that is giving rise to lateral root primordia, but it hasn't been studied very well. And it's like, okay, what are the genes exactly that are contributing there? So we screened a bunch of other genes over there, including the potassium outward rectifiers that we also have observed to have a significant effect on the root system architecture. It's actually preventing the root from growing, which is interesting. We also looked at a bunch of other transporters, and this is umamid, which are basically transporters that are transporting amino acids in and out of the cells. What exactly? Some of them are known to transport glycosinolates, but the specificity of the substrate hasn't been determined yet very well. But we have observed that some of those umamid transporters that are yet to be described are also having an effect on the root system architecture on the salt stress. So this is quite exciting, but this is a very new data. What we have also done from one of our genes that we have mapped that are overlapping between those three different techniques that we have used, one of them is ascorbate peroxidase. So ascorbate peroxidase is basically using vitamin C to deal with the hydrogen peroxide stress. So for that, Mariam has established a collaboration with the lab of Ron Mittler, where we have looked at the accumulation of hydrogen peroxide signal in the plants in our two accessions, the lateral root maker and the lateral root elongator, to see what's happening in response to stress. And what we have observed, and this is, so the technique is really like a visual. You can impose, you can image the plants on the plates and see the ROS signals as you go. So the interesting thing is really that from our lateral root, I think this one is lateral root maker and this one is lateral root elongator, that for one of them, we're seeing, no, sorry, this is a lateral root elongator, this is lateral root maker. Yes. And then for one of them, we see only the signal in the root. For the other one, we see only the ROS signal in the shoot, which again is quite puzzling. Why is that happening? So we are trying to figure out, and again, looking like right now, we are looking at the allelic variation in the ascorbate peroxidase gene between those two accessions to kind of develop hypothesis like what's happening. And then we're going to generate both the knockout mutants and the allele swaps for both of those backgrounds to see if we can reverse this signal. Can we make this accession also have a signal in the root, and how is that contributing to overall root system architecture? What Mariam has also done is looked at how ascorbate itself is contributing to the root development. So in here, we've got accessions. So this graph is very, very busy. So we've got accessions growing under salt conditions, salt with ascorbate, and salt with additional sodium hydrogen peroxide. And what we're seeing is that the accession, let me have a hot second. Okay, so the accession that is a lateral root elongator, so the root, the number of lateral roots is really suffering in here. All of a sudden, we can recover the lateral root making if we add ascorbate to our plates. So ascorbate really seems to be promoting the lateral root making. All right, and then for the other accession, we don't really see much of an effect, but that accession doesn't really have too much problem in the lateral root making. Okay. Again, and this is quite preliminary data. The other thing, so this is the story of like canonical root traits and how they are changing in response to salt stress, which is generating a lot of additional questions. While we were spending hours looking at the plates, at some point I started to really wonder, like see things. And sometimes you see things because it's pandemic, you have been overexposed to root images, you are dreaming that you are a root. But sometimes you see things and it's like, I know, I start to see patterns. And I started to see accessions that were really growing like a Christmas tree, like an inverted Christmas tree, like every child would grow a root. Then you have accessions that are growing like a telephone pole. So all of the lateral roots that are developing, they are growing kind of at the similar length, and they are pretty like 90 degrees. They're not showing too much gravitropism. Then we've got a droopy telephone pole. So the gravitropism kicks in, but at a later point. And then we've got the broomstick. So majority of the roots are really originating from the base. We don't really have too much of the lateral root kind of originating like in the Christmas tree or telephone poles. But when I use my canonical root system architecture traits, like number of lateral roots, average lateral root length, or even the branch zone size, there was always, I got significant differences between all of those like manually classified roots, but I always had one comparison that was not significant, which is like, this is frustrating. I want to combine those traits into one, right? Rather than mapping them individually. So what I did is I started to reaching out to mathematicians, and they're like, yeah, sure, we can totally separate them using Pareto front. And I'm like, okay, great. How did you do that? So Pareto front is a system that people are using to quantify the efficiency of infrastructures within the city limits. So you can have a system, here is a subway network of one of my favorite cities, Amsterdam. And you've got, this is a quite expensive network because you've got all of those different neighborhoods where people are living, affordable housing, and everyone for one reason or another, they really need to get to Central Station. So you can have a network that is very fast, but very expensive, or you can have something that is cheaper, but slow, right? So the expensive, the speed and the cost are typically at the opposite end. So you don't have one optimal solution for your network because you cannot be cheap and fast. Cheap and fast is very dangerous, typically. And in Europe, we're very aware of that. So you want to have something that is either cheap but slow, or you end up with having systems that are fast but very expensive. So we can think about applying the same principles to root system architecture. So we've got those lateral root tips, which a lot of people think like the root tip, that's where a lot of extraction of water, nutrients is happening, right? So these are the affordable neighborhoods that we need to transport all of the resources to. And all of those resources, sure, they need to feed the root system architecture by itself, but majority of them have to go up into the chute. So the chute is our central station. So how can we do that? So if we extrapolate, we can think about that we can connect all of those root tips in a very fast way, in a cheap way, or in an intermediate solution, right? And then basically, so what happens is we've got a front of optimal solutions, and the position on the front is determining what is your prioritization. Do you prioritize the speed or do you prioritize the cost? And then another feature that we've got from this metric is how far from this front of optimal solutions are our roots. And I don't have this slide, unfortunately. But we see that under stress, the plants are really super close to this front of optimal solutions. But as soon as we remove stress, they kind of start getting lazy and they are not optimizing for either speed or cost, which is fair because plants have to think about other things. And at some point, the cost is not really the limiting factor, especially if you're not under stress. You've got all of this photosynthate going. So you might prioritize for the exploration of the soil rather than exploring, like really limiting your costs, right? So, and the good thing is that this trait is heritable. However, our model is not perfect yet. So the main thing that we have not considered is that this speed and the cost is very different between the main root and the lateral root. So in here, I made for you a cross-section of the main root and the cross-section of the lateral roots. Lateral roots, we figured, are very difficult to make cross-sections because they're really thin. But you can see in the blue, you've got the xylem vessels. And the number and the overall surface of the xylem vessel is very, very different between the main root and the lateral root, which affects significantly how the water is being transported. Also, we've got all kinds of changes in the radial water transport, so how the water is kind of getting into the xylem vessel. And also the number of the cortex layers, the size of them is going to also significantly contribute to the cost of the specific root system architecture components. So right now we're collecting, so Emily and Trent have been doing a fantastic job in collecting all of those beautiful pictures that they are quantifying the individual roots, the anatomical components. And we're seeing that we do see changes between the tip of the root and the base of the root for each main root and the lateral root. But the biggest difference we're seeing is that the lateral roots are in general much smaller in all of the anatomical features. So right now we just had a PhD student starting in the lab, Ruth, who is here in the audience. So she's going to take all of those anatomical components, put them into the model, and then we can predict how are those changes actively affecting the water transport and maintenance costs of the specific root system architecture components. And then we can use those updated models, put them into the genome-wide association, map those genes, and really try to understand what does that mean, okay? So another project that I also want to talk very briefly about is this project that we've got supported by the FFAR, and it's a collaboration between my lab, but also a translational lab in the BTI led by Julian Fay, Jim Giovannoni, Silvia Restrepo, and we've got also Myogenics. That's a company here on the campus. It's a Cornell spinoff led by Gagam, and of course, Zangjun is there as well. So in here, what we want to do is we really want to move beyond the genome-wide association within one single species, but really do a pan-genome-wide association. So what we're studying is not a population that is specific to one species, like what we just did, Pimpinellifolium, but we're really looking at the variation across all of those tomato species, which is really cool. And so my lab, we're screening all of those 600 accessions of different tomato species for drought-induced changes. And then the group of Silvia Restrepo is looking at how are those different accessions showing up in terms of resilience to a pathogen Alternaria. So for that, we're really using the drought resilience, it's a very complex trait. So how are we going to kind of divide it into individual phenes? And we're studying only shoots for this specific project. So a first trait that we're going to use is a daily water use. So we're studying that the phenotyping facility has an automated weighing and watering, but with 600 accessions, we're not able to screen all of those 600 into the phenotyping facility. But what we're using instead is this Osmo device. So it's basically an Arduino-based robot. It's all of the details are on the GitHub, but so it's Arduino-based robot that we have a hard-coded information for, like, what is my reference weight for each of my pots? And if my pot is a drought stress treatment, I want my reference weight to be lower, corresponding to 20% water holding capacity. And the plants that we're growing in here are very small. They are 2-week-old plants, so they're not really significantly contributing to my pot weight. So we've got all of the data hard-coded into the Arduino. Arduino is also connected to a scale that we're putting individual pots, not using conveyor belts, but using our digits. And then whenever that reference weight is lower than the hard-coded reference weight on our robot, it activates a little pump and it's doing automatic watering. If we connect this device into our laptop, the data before and after watering is automatically being saved on an Excel sheet, which saves us all of the writing, data transfer, all the problems and mistakes that are coming with that. And it is evapotranspiration, so it's a very dirty trait. But what we're able to see, and in here we've got an example of different cowpea accessions grown under control conditions and drought conditions. So we see that over our average evapotranspiration rate is dropping under drought stress, and that we're also able to identify significant differences between individual accessions, right? And this is only one component. Another thing that we're doing is this, I don't know, it didn't change. What's happening? Is looking at the maintenance of growth. So we are assuming that plants are undergoing all kinds of quiescent events as soon as they realize that they are undergoing drought stress, but at some point they need to restart the growth, even if it's, a little bit. So what we're doing is we devised this very simple pheno cage. So basically everything is constructed using things that you can buy quite easily from Lowe's or Home Depot, wooden cage. And what we're doing is we're putting a plant on this rotating platform. The platform is actually meant for displaying jewelry, but we figured it's also very good in displaying plants. It's turning very slowly, and it allows us to make side view images from 7 different angles. So then even if your plant has a weird posture, you're still able to capture like as much pixels, and this is very highly correlated with the fresh weight of the plant. So we are able to capture non-destructively this digital biomass of the plant and see at which time point, and in here again, we've got drought stress is red, the blue is the control, and this is again, like I'm using cowpea data in here. We're able to identify at which time point in our experiment are we really starting to see differences, significant differences between our plants that are grown with sufficient water and without sufficient water. And then calculate the growth rate out of that, and also relative growth rate. So which plants are performing just as well as under control conditions, and which plants are really suffering relatively. So we're normalizing for all of the differences in the growth rate that we're seeing under control conditions, which is a fact of life when you're looking at the diversity panels. Every one of us grown differently, and if we could only normalize for that, we will be able to learn more. Anyways, so we're collecting those images with the Raspberry Pi camera, and we're using Plant CV to really extract and mask only the pixels that are belonging to a specific plant. Then what we're doing as well is we're looking at plant health indices. So, and we're doing that with using the PhenoSite facility. And if you're interested in using facility, you can always reach out to me. I also left a couple of the stickers that are looking exactly like that next to the coffee machine. I didn't have enough today, so if you want more, let me know. So what are those health indices? And probably people that are working in the field, some of those things might seem familiar, like NDVI, but we're also doing anthocyanin index and chlorophyll index. So for those things, what we're doing is we're moving the plants that are growing in the growth chamber into the phenotyping facility, and we're doing a snapshot measurement. So the plants don't necessarily need to be together growing on the conveyor belt system. We can just use it as a snapshot tool to take the images that we are actually interested in. And here I've got an example of a tomato plant that has been growing in the facility on the conveyor belt, and you can see how the indices are shifting throughout the development. So here are hours of exposure. And again, we've got control and drought stress of the tomato for the chlorophyll index, anthocyanin index, and NDVI. And you can see that the differences that we're seeing are very different for each of those. So, well, chlorophyll and anthocyanin are overlapping to some degree, but NDVI is showing us a very different story, right? And because we've got so many different time points, we're not plotting them with asterisks above anymore. We're starting to plot the minus log of the p-value. So if that line is going above the threshold, it means that the difference is becoming significant. Okay? All right. So that's one aspect of the phenotyping facility traits that we can see, that we cannot really see with our eyes. Another aspect that we can study with the phenotyping facility and the cameras that we've got is the photosynthetic efficiency. So that includes FV over FM, so maximum quantum yield, but also light-adapted quantum yield, non-photochemical quenching. And those are really the traits that are preceding any changes that we're typically seeing on the level of pigmentation. So pigmentation, and I learned that from the people that are selling those machines, the changes in the pigments are very slow to occur. So whenever you see changes like decrease in your chlorophyll index or chlorophyll rate, that plant is already on its way down. So similar, like when I'm going to the beach, my pigments is changing to the red, and the fact that I'm back in here and I'm still red doesn't mean that my relax level is the same as I was lying on the beach. But so the FV over FM, those changes are much more dynamic. So again, we can study that in the phenotyping facility. I'm not sure. So what we're doing, so the typical protocol that we're using is we're doing a dark adaptation of the plant for 15 minutes, and then we're measuring the FV, the minimum fluorescence and the maximum fluorescence, and we're using that to calculate the FV over FM. So this is our dark adapted quantum yield max. Then what we're doing is we're exposing them to the light. We're measuring those same characteristics in the light adapted conditions. And then we can switch the light. And those things, are giving us an information about the non-photochemical quenching and the photochemical quenching. And I have this because I'm a root biologist. I'm not a photosynthetic specialist. So I need that to kind of help myself and remind how those things are calculated. Okay, I do have a graph. So in here, we've got a graph of the FV over FM, FQ/FM, and NPQ. And in that specific scenario, we see that the FV over FM, it took a hot second for it to kick in, but we're seeing significant changes at specific interval during the drought stress. But then the NPQ is showing us a significant difference at a very different time point. So the dynamic aspects of those things is really a cool thing to start exploring. So the phenotyping facility, so here are the plants that are being imaged using the conveyor belt system. So it's allowing us to image up to 1 meter tall plants. So that's 3 feet for those that have problems with metric system. We've got the capacity of 64 large plants. So we can host 64 plants that are as large as you want them to be, up until 1 meter tall. Or if you want to use, so we can switch the tops of the trays that we're using, and then we can have 20 Anaerobidopsis plants that are growing within one tray, which is increasing our total capacity to 1,280 plants. So we've got RGB imaging, and over there, this RGB imaging is much fancier than the RGB imaging that we're doing with our Raspberry Pi, because we're using chlorophyll fluorescence for the masking. So even if we've got plants that have a high accumulation of anthocyanin, we can still accommodate that. So we don't have that the plant is growing, and all of a sudden it's shrinking because it's starting to accumulate anthocyanin. In here, we don't have that problem. As long as there's a chlorophyll, some level of chlorophyll, we can still mask it. We have used it for imaging also of fruits and berries. So it's not really limited only to the plants. And we've got live calculation of very simple plant traits like digital biomass and plant height. And that plant height calculation is very interesting and very important because our chlorophyll fluorescence camera, and you can see it here in the back. So this is the chlorophyll fluorescence camera. Normally those doors are closed, by the way, but for the movie we had it open. So this thing, it's a 200 kilograms machine that can move up and down, and it moves up and down depending on the height of your plant. So our automated plant height calculations are really allowing us to adjust so that your chlorophyll fluorescence and photosynthetic measurements are taken always 50 centimeters from the top of your canopy, which is really important because if that Z axis is fixed, what you've got is a very expensive plant height meter. And so I talked about the different photosynthetic efficiency parameter we can do. We can also develop more protocols if you've got your specific ones that you're interested in. We also have chlorophyll, anthocyanin, and NDVI indexes. And of course, we've got high precision watering and fertilizing, but we can also do any kind of treatment. So if you say, I'm not really interested in fertilizer, I would like to apply champagne. If that's in your grant budget, that's totally fair. But we can do whatever treatment you want. And then, so how do you get to use the Phenosite facility? So we have on the website, we've got a project intake form. So this is really like a starting point for us to make sure that we can accommodate. We're not trying to make false promises or promises that we cannot keep. So this is really for you to describe like, okay, this is the experiment I'm thinking about. I'm not really sure what is possible. I want to apply some like growth promoting bacteria or whatever else. So you fill that out as much as you can. You don't have to fill it out completely, and you send it out to myself. Then we've got the project interview with the BTI greenhouse manager as well, because we want all of the plants to be grown in-house. You don't pay extra for the extra like pre-growing of your plants. And we've got the user agreement with the proposed costs. And yeah, we're growing plants with the dedicated BTI growth chamber. And we can also offer you a snapshot data. So if you've got like, hey, Magda, I've got this plate, or I've plants growing on the plate, or I've got those tomatoes that I just harvested, or apples, or whatever, and I just want to see, is there anything different? We can also do it at the moment. We're doing it free of charge to kind of really offer you a snapshot data, and hopefully with the idea that this snapshot data is going to be so enticing that you're going to come back with a grant money and with the dedicated budget for that. But I don't want to, a lot of people, whenever they're hearing about those phenotyping facilities, they don't have the grant money just yet. So we want to help you out for getting those grants. All right, so yes, okay, yes, I'm finished, or almost. So the take-home messages that I hope to have convinced you somewhat about is that studying stress-resilient plants is really super cool, and we can learn about new strategies that are not present in the plants that we normally are studying. We can use this anatomic or atomic view of the plant phenotype to really combine different strategies that are hopefully complementary and resulting in even more stress-resilient plants. And also sometimes looking at your plants with some imagination and some fantasies can really lead to identification of new ways of quantifying what we're seeing, quantifying those phenotypes. And this is an example of the Pareto front optimality that I talked very, very briefly. I have also presented a series of cost-efficient phenotyping tools. If they are officially also a part of the high-throughput phenotyping facility, but we're not charging for the use of that. So if you're at any point interested in using them or developing those tools yourself, please reach out to me. I'll be happy to help however I can. And also, I have presented some data on the Phenosite. And if you at any point would like to schedule an appointment to come for a tour and tour the facility with your lab members or your colleagues, or just for shits and giggles, you are absolutely welcome to reach out to me, and I'll be happy to show you around my problematic baby. And with that, I would like to thank thank my collaborators, my lab members, and you for your attention, and the organizers for giving me the opportunity to chat to you about those things. Thank you. Yes.
Sorry.
Okay.
As a University of Amsterdam graduate, by the way.
Mhm. Oh, fun.
Yeah. So I enjoyed watching the map. I could see my place of birth later on. Anyway, so the very beginning, you contrasted the two routing structures, more laterals versus less laterals. And then when you showed the transportation map of Amsterdam, you have this like you invest more or you invest less, right? So you inexpensive route architecture and
Fast. Yes.
so, but if you have more lateral, which is the inexpensive one,
Yes.
doesn't it then predetermine for the plant what its growth potential is? If it has, if it's not willing to invest into the roots, it's basically saying, I'm never going to be growing up like
Exactly.
a normal plant.
Yes.
I'm just going to be very conservative, right? Is that?
Yes, you're absolutely right. Yes. So we're calculating the front of optimal solutions for each root system architecture individually, because each lateral root position is different, and it's the front is going to be looking different for the plant with many lateral roots versus with plants with few lateral roots. So those metrics, the Pareto front optimality, is not really designed to look at dynamic systems. So what we're seeing is whenever the plant is growing, the Pareto front, the prioritization, either you are fast or cheap, is going all through, and at some point it stabilizes. So we're thinking it's really like we're not able to kind of capture the dynamic aspect of root system architecture. And also roots are not growing from the tip to the root, they're growing from the root to the tip, right? So you're absolutely spot on, and we are trying to develop models that are better in capturing those like dynamic aspects of that. Yes.
So from a breeding perspective, then, would you want crops when they are salt tolerant? Would you want them to have then that inexpensive root architecture or not? What would you be breeding for?
So I would say from the breeding perspective, and again, like it's we have even on the very simple traits like number of lateral roots, we've got genes that are contributing differently to the overall salt tolerance. So we've got genes that are like SOS1, which is like making the plant super salt tolerant. Those plants are making more lateral roots. But we also recently have identified a DUF247, which is involved in the suberization of the roots. And those plants are also having lower number of lateral roots, but they are more salt tolerant. So that's why I'm like, the relationship between a trait and the tolerance is very, very complicated story, because it's really dependent on the molecular context. And that's why, so we're trying to really understand, like, what do we want? From when we're pushing the root system architecture from one architecture to another using hormonal applications, so we are able to push, for example, a Christmas tree to become a telephone pole by applying auxin, because that's a typical auxin phenotype, right? So when we're doing that, what we're seeing is that the plants are typically accumulating much more sodium ions, because all of those new lateral roots need to make space in their suberized layers to kind of for all of those lateral roots to emerge, which they are patched, but they're not patched perfectly. So that's causing additional leakages into the root system architecture. But that's an auxin story, and that's probably going to be working under specific conditions. And I think what we are moving slowly, it's away from like one ideotype to an emergent properties of a system that need to be, all of the ifs and ends need to be in place to really move the plant to a different level of tolerance.
Well, I am afraid we are out of official time.
Sorry.
Thank you again, and if any of you have questions for Magda, I'm
Yes.
sure she'd be happy to chat. Let's try again.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.